Hello everyone. Thank you for your interest in this overview of AAV. In this introduction to AAV mediated gene delivery, I will provide a summary of commonly used virus-mediated gene delivery solutions, highlighting their strengths and weaknesses. I will explain how AAV works in gene therapy, describe AAV tropism, present the AAV therapies that have been approved by the FDA, and outline the process of AAV particle production. I hope you find this introduction and overview of AAV helpful. What if we could use viruses not to cause disease, but to cure it? That question is not science fiction, it's science right now. I'm thrilled to have you here for a conversation that might change the way you think about medicine, biology, and even the viruses we've long feared. When we hear the word virus, most of us think of illness, outbreaks, and pandemics. But what if I told you that those very same viruses could be reprogrammed, redesigned, and repurposed, not to make us sick, but to heal us? Today, we're diving into the cutting edge of viral therapeutics, how researchers are turning one of humanity's oldest enemies into one of our most promising allies, from targeting cancer cells with precision to correcting genetic disorders at their source. Viruses are being engineered to act more like microscopic heroes than villains. Whether you're a scientist, a healthcare professional, a student, or simply someone curious about where the future of medicine is headed, this session is for you. So get ready to explore the science, the challenges, the breakthroughs, and the possibilities. Let's begin. When it comes to viral gene delivery, AAV isn't the only option, though it's certainly one of the most popular. Different viral vectors each have their own strengths, and understanding these options is key when choosing the right tool for the job. Here are the major viral vector types used in gene delivery. And I'm going to start with adenovirus. So adenovirus is known for their high transduction efficiency in both dividing and non-dividing cells. These vectors excel at delivering large genetic payloads, but they can provoke stronger immune responses, which limits their long-term use in therapies. Lentivirus, which is part of the retrovirus family, lentiviral vectors are powerful tools for integrating genes directly into the host genome. They're especially valuable for stable gene expression and often used in CAR T cell therapy and stem cell application. Lentivirus gene delivery is actually a method we focus a lot on at OriGene because of its broad utility in clinical research. Along with lentiplasmids and lentiparticles, we also provide a number of useful lentiaccessories to support researchers using this method. HSV, or herpes simplex virus. This vector, along with having the highest carrying capacity, is also particularly effective at targeting neurons, making them a strong candidate for neurological research and gene therapy in the brain. And then let's return to the top of the table in the star of our webinar, AAV. Now, AAV's ability to deliver genes with long-term expression and minimal immune response makes it ideal for a wide range of therapeutic applications. Although the carrying capacity for AAV can be rather limiting, the broad tropism makes AAV one of the most versatile and efficient delivery methods. As you can see, each of these viral vector types have distinct advantages depending on the target tissue, desired gene expression duration, and immune response concerns. This allows researchers to choose based on the specific needs of the therapy. To understand why AAV is so powerful, let's start with how it works. And the first step is vector engineering, which means researchers modify the AAV genome by removing its viral genes and replacing them with a therapeutic gene or your gene of interest. This customized AAV can no longer replicate, but it's still highly effective at delivering genetic instructions directly into target cells. So what happens next? Once administered through injection or infusion, the AAV particles locate the target cells and they bind to specific receptors and enter the cell via endocytosis. Once inside the cell, the AAV escapes the endosome and enters the nucleus. Unlike some other viral vectors, AAV doesn't integrate into the host genome. Instead, it forms a stable episome, which reduces the risk of unwanted mutations. This unique feature makes AAV especially safe for long-term gene expression, a critical factor for therapies that require consistent protein production. Here's what's remarkable about AAV, its versatility. Each AAV serotype behaves a little differently, giving scientists precise control over which tissue the therapy targets. In the table that I put on the slide, I'm showing you 13 different AAV serotypes, and then listing alongside of them the corresponding tissues that they have the greatest affinity for, and then some relevant applications each serotype is used for. Highlighting just a few of these, AAV2, which is one of the more widely used serotype, is fantastic for retinal delivery and is used widely in applications related to retinal disorders. AAV5 has an affinity for liver cells, so it's ideally used for hemophilia research and treatments. AAV9 is I would consider a standout AAV serotype because for its unique ability to cross the blood-brain barrier. This makes it a perfect vehicle for studying neurological diseases. This flexibility allows researchers to match the right serotype with the right condition, increasing treatment success rates. I can only imagine how exciting it is for researchers to have a tool with this type of specificity and unique tropism. Now, how does this look like in real world medicine? Let's talk about some groundbreaking therapies that are already changing lives. So AAV is more than just a theory. It's saving lives right now, and I share with you a few of the FDA-approved gene therapies. The first of which was approved in 2017, Luxturna, which uses AAV2 and is being used to restore vision for patients with inherited retinal dystrophy. One of the exciting aspects of all these FDA-approved AAV therapies is the success that has been demonstrated using a variety of different serotypes. I already mentioned AAV2 to target retinal disease, but we also have AAV9 to target spinal muscular atrophy, and you have Roctavian, which uses AAV5 to help hemophilia A patients reduce uncontrolled bleeding through stable factor 8 expression. These therapies are truly remarkable, and with more in the pipeline, AAV's potential in medicine will only continue to grow. And finally, let's look at the process for producing AAV particles, or the viral vectors that contain the genetic material to be delivered to target cells. The process starts with plasmid design and preparation. In the standard method of producing AAV particles, 3 plasmids are utilized. The transgene plasmid carries the therapeutic gene. The helper plasmid provides the viral functions needed for replication. And then the RepCap plasmid encodes the replication in capsid proteins. The 3 plasmids together are co-transfected into what are called producer cells. HEC293 cells are commonly used here. You can think of them as molecular factories designed to generate AAV particles. Once transfected, these cells begin producing AAV capsids, which accumulate over the next 48 to 72 hours. The viral particles are released into a crude lysate when the producer cells are lysed. To measure titer, researchers can utilize methods to measure intact capsid. OriGene has available AAV ELISA kits that use recombinant monoclonal antibodies specific for serotypes. Another method for measuring titer is qPCR. OriGene's AAV qPCR fast titer kit can be used to measure genome copies and is serotype agnostic. At this point in the production process, if you find that your viral titer is low, it is possible to rescue low titer viral particles. OriGene's AAV concentrator can rescue low viral titer by as much as 100-fold. Purification of AAV is crucial. Methods like cesium chloride gradients or affinity chromatography help isolate high purity viral particles. Finally, rigorous tests include qPCR, Western blotting, and ELISA assays confirm the safety and potency of the vector batch. For researchers who choose, companies like OriGene provide pre-made AAV ORF particles. At OriGene, we have over 30,000 target-specific AAV particles, enabling researchers to study a wide range of genes across different species. And this concludes my presentation and overview of AAV viral-mediated gene delivery. I thank you for your time and encourage you to visit origene.com to learn more about AAV and other virus-mediated gene delivery methods.